We are presenting our experiment on the comparison of varying food sources on the attraction of Drosophila melanogaster.
I'm Reed Anderson.
I'm Hayden Flume.
I'm Matthew Giddens.
And I'm Josh Goforth.
Fruit flies, formerly known as Drosophila melanogaster, originated from Southeast Asia. The Drosophila melanogaster migrated across the world to places such as North America, North Africa, and the Caribbean. Drosophila are a nuisance to the ordinary person due to their desire for overripened fruits, which lead to problems for farmers and grocery stores. As a result of the invasions and complications caused by the Drosophila, regulations and inspections have been created.
The life cycle of the Drosophila melanogaster begins with the production of the ova in the female ovary, which takes around 3.5 days. After that, once fertilization has occurred, the larva takes around 24 hours to hatch. And after the larva is hatched, each subsequent repetition of the shedding of the exoskeleton is known as an instar larvae. So you have your first, your second, and then by the time you're third is when you enter the pupa stage of the life cycle. From there, the pupa enters adulthood and becomes an adult Drosophila melanogaster. Drosophila melanogaster are used commonly in science due to their short life cycle, fast reproduction time, and wide availability of genetic mutations for purchase. Also, scientists have lots of prior knowledge regarding the species in genetics, psychology, behavior, and ecology. These are also found in homes, so they're very common, and that is why we use them in this experiment. Our independent variable is the different food sources that we chose to provide a wide range of sugar concentration, and these vary between 0% sugar, which is our control, 1% sugar in the potato, roughly 20% sugar in the banana, and roughly 80% sugar in the honey. And this was in order to mimic the best natural food sources for the fruit flies. This application of our IV can be utilized in organizing the home, and agriculture, and in scientific general knowledge of fruit fly behavior.
The purpose of our experiment is to determine the favorable food source of the species Drosophila melanogaster. In addition, the experiment will be an effective way to increase laboratory experimentation skills, practice analyzing and reporting data, and learn about Drosophila melanogaster behavior.
The hypothesis of our experiment is if the male and female Drosophila melanogaster are exposed to varying food sources like the potato, banana, honey, and control, as discussed before, then the male Drosophila melanogaster will have the fastest and highest completion time of the maize we build, with honey as their favored food source due to its highest sugar concentration.
The independent variable of our experiment was the different food sources, which contain varying sugar concentrations. The dependent variables of our experiment were the average time of completion of the maze and the percent of flies that completed the maze. Our control was running a timed trial that had no food source at the end of the maze.
The procedures for our experiment roughly follow these. Before any experimentation took place, we received our flies from Mr. Atkins, and we allowed time for reproduction before we prepared for our experiment. After 5 days, the vial of flies and pupa were placed in the freezer for 3 minutes to anesthetize. We examined the flies through a magnifying glass and split them into male and female populations. Each population was then split again to form 4 populations, 2 of each gender. Prior to experimentation, I created a wooden maze shown in this slide. The maze had 2 removable cubes that could be sealed separately to the maze. These were our beginning and ending locations for the maze. For the trial portions, as stated before, we used potato, banana, and honey, as well as an empty trial as the control. The appropriate food sources placed in the exit box, and the flies were anesthetized and placed in the entrance box. Once the flies awoke, the door to the entrance box was opened and data collection began. As for data collection, for each trial, time was started once the entrance box was opened. Time was recorded each time a fly reached the food source. The data was recorded in the appropriate area on our data sheet. Once all 4 trials for all 4 food sources were complete, the percent completion for every single trial was calculated by calculating the completed amount of flies divided by the total amount of flies in that trial multiplied by 100. Additionally, the average time of completion for each trial was calculated using each completion time added up and divided by the number of flies that completed that trial.
The graph above displays the percent completion of the flies in each of the 4 trials of the maze. The first trial consisted of no food at the end of the maze, which resulted in no flies completing the maze in both males and females. The second trial consisted of a potato at the end of the maze, in which 6% of the males on average completed the maze, while around 10% of the females completed the maze, which was around almost 4% greater than the males. The fourth trial consisted of banana at the end of the maze, in which 22% of the males completed the maze, whilst around 16% of the females completed the maze. The females in this trial completed on average 6% greater than the females did in the potato trial, and the males completed around 16% greater than the males in the potato trial as well. The males in this trial did complete the maze around 6% greater than the females in addition. The fourth and final trial consisted of honey at the end of the maze, in which 6% on average of the males completed the maze, while around 11% of the females completed this maze. The males in this trial completed around 16% less than the males in the banana trial, and the females in this trial completed around 6% less than the females in the banana trial. However, the females in this trial completed around 4% greater than the males in this trial. The overall trend points to the banana trial as being the most completed maze of the flies that participated in this trial. The next graph displayed above displays the average time of completion of the flies that did complete the maze in each of the 4 trials. The first trial again contained no food, in which no flies completed the maze, therefore there was no average time of completion. The next trial again consisted of the potato at the end of the maze, in which the males completed the maze on average around 290 seconds, while the females finished the maze around 287 seconds, which results in the females being around 3 seconds faster than the males. However, that was not significant in our findings. The third trial consisted of the banana at the end of the maze again. The males completed the maze on an average time of 184 seconds, which was 106 seconds faster than they did in the potato trial. The females in the banana trial did complete the maze in around 200 seconds, which is 86 seconds faster than the females in the potato trial. However, 16 seconds slower than the males in this particular trial. The last trial again consisted of the honey, in which the males completed the maze around 291 seconds, which was 170 seconds slower than the males in the banana trial, and the females completed around 260 seconds, which was 31 seconds faster than the males, but nearly a minute slower than the females in the banana trial. The overall trend points to the banana trial as having the fastest flies complete the maze, and of all the 4 trials.
Our conclusion was that the data did not support the hypothesis that the male Drosophila melanogaster exposed to honey with a sugar concentration of 82.12% would have the fastest and highest percent completion of the maze. Instead, the male Drosophila melanogaster had the fastest and highest percent completion of the maze with the banana as the food source, with a sugar concentration of 20% by weight of starch.
The graph above again displays the percent completion of the flies in each of the 4 trials. It was predicted that honey would have the highest percent completion of the flies due to its high sugar concentration of around 82%, which was significantly higher than the rest of the food sources in the trials. The honey, however, did not have the highest percent completion and was actually less than the banana trial, even though the honey had a greater sugar concentration. The banana had a sugar concentration around 20%, which was nearly 62% less than the honey trial. Again, this graph displays the average time of completion of the flies that did complete the maze. In addition, the flies in the banana trial were significantly faster than the flies in the honey trial, even though that the honey trial had a significantly higher sugar concentration than the banana. The honey was predicted to be the fastest due to its high sugar concentration, but was later determined that it would not have the fastest Drosophila melanogaster. In addition, the flies in the honey trial were slower than the flies in the potato trial of the males, by only 1 second, which wasn't significant for our data collection, but did raise a question for us due to the significant sugar concentration difference between the two. After reviewing various scholarly journals and resources, it was determined that the natural food source is an overripened fruit or decaying fruit for Drosophila melanogaster. Their choice of food, however, was determined that it isn't entirely due to their digestive and nutritional needs. Their food source is also their site for reproduction and housing of eggs in conjunction with their source of nutrition. Although the honey had a higher sugar concentration, its viscous character was not optimal for reproduction. However, the banana had a significant sugar concentration and was able to house the eggs of Drosophila melanogaster. Therefore, the most desired source of Drosophila melanogaster isn't directly dependent on the sugar concentration of food, but rather the combination of multiple factors such as sugar concentration and ability to house eggs following reproduction.
In a previous study that we looked at regarding our same variables, there was a fruit fly study in Brazil due to the large amount of fruit loss in their agricultural fields, and the manipulated variable was also food sources, as they were trying to determine which food source the fruit flies migrated to the most in order to minimize their fruit fly infestation in their fields. They separated the actual food sources into 4 different fields and determined which one the flies were most attracted to.
Improvements that can be made for our experiment are relatively brief, however, they could improve our data. First, a perfect seal at the entrance of the maze could be made because a few flies were able to escape through small gaps at the beginnings of our trials. We could have done this by not having removable entrances and exits, or by sealing the joints carefully before every trial. Additionally, the flies could be allowed more time to reproduce in order to obtain a larger sample population. While our experiment was successful, it would be more reliable if a larger sample size was used.
One of the obvious issues in our experiment that we noted was that the fruit flies were more attracted to something in their natural environment. So for a future experiment, our food source, instead of being our control, potato, banana, and honey, could be organic and conventionally grown bananas, to determine if the level of attraction of fruit flies to their natural environment is greater, or if it's just the food source itself. And to do this, we would use basically our same procedures, except with our modifications talked about previously, and we would determine whether the organic or the conventionally grown bananas are ideal for fruit flies.
Thank you.
Thank you.
Thank you.
Thank you.